Okay, let's do genetics. Now, genetic study comprises of the basic molecules, chromosomes, which are made up of genes, and DNA, which are made up of proteins, and two helicals, which are negatively charged and bound around each other by positively charged proteins, which are called as histones. So that's all the basic structure of DNA, which in turn interpret the genes and which in turn form the chromosomes. Okay, this is all where our genetics is moving around. Now, looking into the genetics, we need to know why genetic testing is carried out. So the first thing genetic testing is carried out is to look for carriers. Why do we need to look for carriers? Is sometimes we have a high degree of suspicion, or like if we want to find out if a person is carrying a gene, uh, like in cases when already one partner is carrying a gene and you want to know if the other partner is also carrying a gene, otherwise both of them are carrying it, there is a higher chances of the baby getting affected with the disease, right? So that's why you need to carry out a testing in those people, that is known as carrier screening. Then the second time it is done is for prenatal diagnosis. So prenatal diagnosis, it has become a part because it is, we need to know if the baby that is developing normally or not, the baby that will be born will have any genetic diseases or not. So for that, prenatal diagnosis is carried out. What are the methods by which you can do prenatal diagnosis? The first is amniocentesis. So what happens in amniocentesis is a fluid is taken from the mother's belly, from the amniotic cavity in which the fetus is developing, and the fluid undergoes all the testing procedure. So you study all the chromosomal structures of the fetus from the fluid and you come to know if anything is going wrong or everything is proper or not. Okay, that is amniocentesis. Second is chorionic villus sampling, which is generally carried out at a bit later stage. What happens over here is the umbilical cord, it is attached to the placenta in a mother, right? So the placenta has chorionic villi in it. From that area a biopsy like is taken, and from that the study is carried out to look for the genetic diseases. Okay, that is chorionic villus sampling. Now sometimes pre-implantation genetic diagnosis is generally done when a person is having in vitro fertilization. Those couples which have fertility issues, they undergo in vitro fertilization. So the embryo is literally developed outside the body and then implanted inside the mother to develop. So in those cases, you it is better if we know if the fetus is having any chromosomal abnormality or not, right? Otherwise, if the fetus is having abnormality and the mother is undergoing so much procedure and in the end result the baby comes out with some or the other genetic defect, it's going to be problem, right? So that's why you do a pre-implantation genetic diagnosis. Then fourth one is a new method. It is an analysis of fetal DNA in the maternal circulation. It's a kind of a newly invented method where blood sample from the mother is taken, and in that blood sample we look for the DNA of the fetus that is found out, and that DNA is studied to see if any genetic problem or diseases is there or not. That testing is called as analysis of fetal DNA in maternal circulation. So these are all the parts of prenatal diagnosis. Before the child is born, you want to know if everything is healthy or not, right? Next, we do the genetic testing is in pre-symptomatic diagnosis. So when we know that a person has a family history of a disease, and to know if the person is going to develop the disease or not, so you can test for the gene. So the person is carrying the gene or not, like what Angelina Jolie has done, right? Her mother had breast cancer, so the defective genes are BRCA1 and BRCA2. We have read so much about it. So she underwent a testing for BRCA1 and BRCA2 genes, and if the full form of BRCA is breast cancer antigen, right? So if when you look for those antigens and they were positive in her, and so she underwent a bilateral mastectomy to prevent getting breast cancer. So the testing when she got that BRCA1 gene and BRCA2 gene tested in herself, that was a pre-symptomatic diagnosis. Okay. The fourth scenario where you need to do genetic testing is when you are testing if the drug that you are giving to the patient is efficacious or sensitive or not. Is the drug working or not? So that is, again, I'll give you a breast cancer example. breast cancers do generally have estrogen and progesterone receptors on their surface. Now there are drugs which are working against these receptors, so you need to know if the estrogen and progesterone receptors are present in the breast cancer tumor or not. If they were present, then only your drug is going to be effective, right? If the receptors are not present, the drug is not going to be effective. So that's why you need to do the testing for the drug efficacy or sensitivity. And these are the scenarios when it is a need to do a genetic testing in an individual. Now coming from that to a new method that was invented, it is called as gene therapy. Gene therapy is very in every lecture I have included a topic which you will feel like, okay, why do we need to know that? That is because to help increase your knowledge, because these are the topics which are like happening around. Okay, these are like the latest things that are going around in that field. So one of them in this is gene therapy. So what is gene therapy? Now some diseases, what happens is there is a total the cells are there, but there is a deficiency. They are not producing enough of the enzyme or enough of the substance that is needed, the chemical that is needed in the body. So the scientists got together and they developed a method so that they could put the gene inside the human being, so the deficient enzyme could be produced. That method is called as a gene therapy. So what do you do in a gene therapy is you have a vector which will carry the normal amount of the gene, and the vector is inserted into the affected individual. The vector is generally a virus, okay, not a pathogenic virus, of course you don't want to cause a disease to the person, a virus which is not going to be active, that is genetically modified. They put the gene, the normal gene, in the virus so that the virus will go inside the human body, and it will like HIV virus implants itself like that, this virus will also implant itself in the human body and it will multiply. As it multiplies, the gene copies will start developing into the individual, and as the individual starts developing the gene copies, the deficient gene will be filled up for, and the person will be able to make the enzyme that it was deficient in. So this has been carried out in one of the disease which is a immunodeficient condition, where the individual has a deficiency of enzyme known as adenosine deaminase. That is an enzyme which is present in the T cells, which helps us fight against all those viruses and bacteria and pathogens. So what they did is they developed a vector who could carry the gene which was coding for this enzyme, and they put it into the individual who was immunodeficient. So the vector went inside the human being and it multiplied itself, and the enzyme and the gene got into the individual, and the gene started transcribing the protein, and the enzyme started forming, and that is adenosine deaminase. And so the person who was immunodeficient could give some immune responses, so it started developing some immunity, right? So that is how the gene therapy has started coming into the use. Okay, for it can encode for the missing gene in the individual. Hopefully gene therapy progresses properly and you can code for all the major diseases so that people can get cured. That's why it's a topic much to be discussed. Now coming to the basic structure of DNA, we know there are two strands which are helices, we call them. They are wound around each other. They have two ends, the 5' end and a 3' end, and they are like they are opposite to each other, like this 5' end over here, the 3' end over comes over here, and here if this one has a 3' end, the 5' end of the other strand comes over here, right? And what are the base pairs which are on the two strands? They are the cytosine, which will bind with the guanine, and adenine, which binds with the thymine. Okay, these AT bind with 2 bonds and CG bind with 3 bonds. They are all lying on a chain which is tied with each other by a phosphodiester bond. Okay, this is too much knowledge, it's okay if you don't remember, you just need to know this pairing. You definitely need to know the pairing where C goes with G and A goes with T always. So if it's a here, there will be a T here, but if there is a T here, there will be a here always. If there is a C here, there will be a G here, and if there is a G here, there is a C here. That's how it goes. Okay, but this is for DNA. In RNA, the only difference is same bonding, except instead of A, you have uracil. Okay, so U goes with T. That's the only difference in RNA. Okay, now Now what are the some codons? They all make a codon, like you saw this C, G, A, T. There are some codons when they three of them come together, like here you see this C, A, G is there, or if this one is from the above one, then you can say A, G, T is getting formed, like that. Then this is T, C, A is getting formed, right? So there are these codons like U, A, G, U, G, A and U, A, A. They are called as nonsense codon. Why nonsense? Because as soon as these codons come, these three base pairs come, the transcription process stops. Whatever process is going on, transcription or protein formation or any process that is going on in the body comes to a stop. So these codons stop the process. They are called as nonsense or stop codons. This is the information replication. What are the complementary base pairing? A with T and C with G. Then the enzyme we have to know about is known as the DNA polymerase enzyme. Why is it important? Because it helps keep both the strands separate when they are getting replicated, because you need to, these are two together, right? But you separate them and you form new strands parallel to this and parallel to this one, so that to form the two strands parallel to them, they have to be kept separate, and that is kept separate by DNA polymerase enzyme. Not only does it keep separate, it also keeps a check that the new pair that is getting formed, that it is getting replicated, should be proper. Nothing should go wrong in its replication. Okay, that is DNA polymerase enzyme. Now coming to the mutations, the genetic disorders. What are mutations? Mutations are the inherited alterations of the genetic material. And what are the types? They are base pair substitution, where one base pair, what is a base pair? I again told you just now, what is a base pair? This is called one base pair. Okay, UAG, UGA, UA, these are all base pairs. That one base pair is replaced by another. That is called as a base pair substitution. When the substitution changes the amino acid which it is going to transcribe for, it is known as missense mutation. And when the substitution produces any of the three stop codons, the UA, UGA, UA, then it is called as a nonsense mutation. Why? Because it will stop the process, right? So what happens is these base pairs, they are coding for a specific amino acid. You know, there are like plenty of amino acids for the body. So all these base pairs are coding for the amino acids. Now if only one base pair is changed and the structure of amino acid changes, that is known as missense. You misunderstood somebody, you can again clarify it, you can again correct it, then it is missense. But if you produce any of these nonsense codons, you stop the procedure, so it is a nonsense. Oh my god, you stopped it, so it's a nonsense mutation. That's how I tried to remember it. I'm just helping you. The other kind of mutation is known as frame shift mutation, when there is an either insertion or a deletion of one or more base pairs. Okay, so either one of the base pair will get inserted or one of them will get deleted. That is known as frame shift mutation. That was about the types of mutations, just the names of the types of mutation. All these mutations can cause one or the other genetic diseases. Okay, but I'm not going into so much detail, otherwise it will be a lot of genetic diseases to know about. Now this is knowing about the basic genetic processes that are going on in our body. What is transcription? It is the formation of the RNA strand from the DNA. And what is translation? It is the formation of the protein strand from the RNA. Okay, so what happens is the DNA strands, they get, in the process of transcription, they get separated and a complementary base pair is formed. That complementary not base pair but a complementary strand, that complementary strand is called as a RNA, and that RNA comes out and it can translate itself into a protein formation. How does the protein get formed? This whole RNA will form codons which will give amino acids, and all the amino acids will be formed according to the codons, and that amino acids will get together and form a protein. That is how the whole translation process goes on. Okay, now when this RNA is getting formed from the DNA, what happens is these all codons are there, right? So some of them codons remain on the strand while some of them are let loose. That procedure is called as RNA splicing. So the ones which are excised, which are taken out from the strand, are called as introns. And introns are basically, they can do all the other work, like keeping a check if the procedures are going on properly or not. Sometimes those introns can go crazy and cause mutations also. So they basically are the excised sequences. They are not doing the work that they are supposed to do, they are doing all the other work. Whereas the sequences that are left on the RNA strand, which are going to code for the amino acids, those are called as exons. Okay, and they code for the amino acids which will in turn form the protein. Okay, so those are called as exons. That was the RNA splicing procedure. Now what are human cells types? Human cells are classified into two types: the germline one, from which the other cells are rising, and the somatic cells one, which are all the other cells. Okay, now mutations, of course, because these are germline cells, you know, the examples are sperm and the egg cells. Whenever there are mutations in the germline cells, they are going to be transmitted into the next generation, right? Because you are developing a fetus from those cells. But in the somatic cells, if any mutation occurs, it generally does not go to the next generation. Okay, now what are gametes? Like these sperm and egg cells, the other name for sperm and egg cells is gametes. Okay, they are the haploid cells. And why are they called haploid? Because all the other cells of the body contain 22 pairs of chromosome, plus if it is a female XX, if it is a male XY. But gametes, like these sperm and egg cells, they are called haploid because they will only contain 22 chromosomes, not 22 pairs. Okay, only 22 chromosomes plus either X or XY, either X or Y, sorry, either X or Y. Okay, only one of the chromosome. Okay, so that's why they are called haploid cells. And why they are like that? Because they combine with the other, right? Because a sperm combines with X cell, so it will have 22 plus, a sperm will have 22 plus X or 22 plus Y, and the X cell will have 22 plus X. Always, it always has an X. So they both will combine and they will form 22 pairs of chromosome plus whichever is coming from sperm. If it is coming X, then X and X will form a female, and if it is coming Y, then it will be X and Y getting together forming a male. Okay, so that is why the gametes are always haploid. And how do they become haploid? How do they become half? The process is called as meiosis. Okay, where you go the division twice, that is meiosis. So the diploid cells become haploid, half. Now why we discuss that basic is cells that have, so the normal number of chromosomes in a human is 22 pairs, plus female is XX and male is XY. So they should be 23 pairs. Okay, but if they have a multiple of the normal number of chromosomes, like normal 23 pairs, then they are called as euploid cells. When this euploid cell has more than the diploid number of chromosomes, like, sorry, it should not be euploid, but when a cell has more than like more than 23 pairs, it is called as a polyploid cell. And when it has 3 copies of each chromosome, like it happens in Down syndrome, so instead of two pairs at all the places, one of them can have 3 copies, then it is called a triploidy. When aneuploid is called when they do not have in a multiples of 23, right? So the chromosome should be 23 pairs, that is 23 into 2 is equal to 46 chromosomes. But instead of that, there are only 45, or there are only 44, or there are only 40 chromosomes. Then they are called as aneuploid cells. Okay, aneuploid is usually the result of non-disjunction. You can say why there are less, because when they were supposed to get separated, they did not get separated. They stayed joined together. In the process of meiosis and mitosis, they stayed together, and so they did not get separated. That is called as non-disjunction. And so because of that, there are homologous chromosomes or sister chromatids which they fail to separate, and so they are joined together at that place, and so the counting did not come out to 46, it comes out to either 45 or 44 or something like that. Okay, so that's why that is an aneuploidy. This is an example showing how did the aneuploidy happen. So what normally should have happened, these two get separated, and they, one goes here, one should ideally, like here they are showing, one goes here and one goes there. Ideally this strand should also have separated, one going here, one going there, and this one also, one going here and one going there, right? But when a non-disjunction happened here, the non-disjunction happened over here only, so there, that's why there are two here, so two pairs over here, and you see over here this is with the fertilization with a normal gamete, so one pair came from here, and you can see over here trisomy is formed. Or maybe there was an empty one over here because there was none here, so a monosomy was formed because only one pair came from, one haploid came from there, right? So monosomy resulted. Same here, non-disjunction occurred over here, so there were two pairs over here. Instead of one and one going, there are two. So again, a trisomy or a monosomy can happen. Okay, so that is how aneuploidy number of cells can happen. The best example of aneuploidy is Down syndrome. You see over here at the 21 chromosome, there are 3 pairs. Instead of 2, there are 3 pairs of chromosomes. So, and that is called as Down syndrome. What are the symptoms that you will see, or the signs that you can see in a child affected with Down syndrome? Growth failure, mental retardation, there is a flat head, the ears are at an abnormal place, the child will have a small hands and broad hands, and instead of so many creases, if you look at your hand, you all will have like 3-4 creases in your hand, right? But Down syndrome will only have one crease in it. It's called a simian crease. It's quite common, so only one crease in the hand. Now that's called a simian or a palm crease. There will be skin ridges on the baby. There will be there might be absence of a rib. There might be any intestinal blockage. There can be umbilical hernia. The pelvis can be abnormal. The muscle tone will be very diminished, like hypertonia or hypotonia. There can be other abnormalities also, like toes will be very widely spaced. The child can also have some or the other congenital heart diseases. It can also have the colon problems. So it is basically having problems everywhere. You know why? Because in the fetal stage only the problem has happened, right? So all the other organs developing after the birth are going to have some or the other problems, right? Complications. So that is Down syndrome. Turner syndrome, you can see over here, there should have been 2 X chromosomes, right? Because it's a female, but instead there is only one. So how do you say the genotype of a Turner syndrome? 22 plus XO. Normally it should be 22 plus XX for females, or 22 plus XY for males. That's the normal genotype. But in Turner's it will be 22 plus XO because one X is missing, again due to non-disjunction. Okay, what are the features you will see in a Turner syndrome female? Is a short stature, a low hairline. There will be the shape of the thorax will be like a shield. It will be protruding outside. The nipples will be widely spaced. There can be brown spots on the skin. The female, because one X is missing, so the genital organs are not developed properly, so she will not have any menstruation. Because the ovaries are not developed, the uterus is not properly developed, so all the reproductive tract is not properly developed, she cannot conceive. There will be other elbow deformities. There will be other complications like heart aorta problems, and there will be different facial features. Those other features, again as I said, like in Down also, it was because it is from the fetal stage, right? So same with the Turners, it is from the fetal stage, so many deformities are developing all over the body. Another example is of a Klinefelter syndrome. So you see over here, it is a man, he should have X and a Y, but instead it has 2X. Why? Because a non-disjunction happened, and so instead of 1X coming, it 2 X's has come. Okay, so it's a XXY. The genotype of a Klinefelter syndrome is 22 plus XXY. What are the features of the individual? The person will be taller. There will be less hair on the face, less hair on the body. The person, because of 2X chromosome, is going to develop some female characteristics like breast development, and there will be female type of fat distribution. And because there is X is so dominant, the Y will not be able to work, and so the testes will not be developed in the individual. So even a Klinefelter syndrome can have infertility issues like that. Okay, so that's all examples of that's all the features of a Klinefelter syndrome. Now, that was one kind of disorder we saw, genetic, where you had an abnormal number of chromosomes. Now here what happens is the material of the chromosome gets changed. So sometimes what happens is the chromosome has a very definite structure. Okay, believe it or not, microscopically every place of the chromosome is defined, that there is a gene present over there which encodes for specific things, specific proteins, specific enzymes for the human body. Sometimes what can happen is a break can happen in the chromosome and the segment of chromosome will flip around. Okay, so a fragment comes out from here, it flips around and gets attaches again back over there, but in an inverted order. Now what happened over here is the chromosome is balanced because the fragment did not come out, you did not lose the fragment, right? So it is balanced, only that it is changed, the structure has changed. Okay, so that's a balanced alteration of chromosome. What happens sometime is the portion comes out from this strand and it will go and attach itself to another chromosome, and a portion from that chromosome will come out and attach itself to this chromosome. That is known as translocation. So two chromosomes, like it happens between chromosome number 9 and 22. So it's a very common example, it is called as T9 comma 22. So what happens is a translocation, portion of the 9 chromosome came out and attached itself to the 22 chromosome, and the 22 chromosome portion comes out and attaches itself to the 9 chromosome, which is generally seen in leukemia, CML, chronic myeloid leukemia. So it's a very common translocation. Okay, so that is what is known as translocation, portions getting exchanged between two types of chromosomes. One example of a translocation is a Robertsonian translocation, where the long arms of two non-homologous chromosomes fuse and they form a single chromosome. What happens over here is they get joined together and they form one chromosome instead of two. That has seen in cases in chromosome 13, 14, 15, 21 and 22. Another is reciprocal translocation. So it occurs when break takes place in two different chromosomes and the material is exchanged. That is just what I explained an example, that the translocation between 9 and 22, the chromosome fragments got broken and they got exchanged. Okay, so that's a reciprocal translocation, type of translocation. Okay, one of them is Robertsonian, the other is reciprocal. So this is just an example to show you the chromosome structure abnormalities. Here you see this is lost, so it will become like this. That is a deletion example, a base pair is lost. Here the base pair has got duplicated, right? So then here you can see over here B and B again forming, and again it is developing, so it got duplicated. That is a gain. Now balanced is when this happens. You can see over here, instead of B coming here, it went and attached itself here, and the D from there came here. See, you can see over here the D is coming here and the B is going in its place, so they exchange places. No loss, no gain. Okay, and reciprocal is, you can see over here, right? So they changed. You can see in the picture, I know my timer is coming in between, but here you can see, right, this is getting translocated, the portions are getting translocated. Now Philadelphia chromosome. What happens in Philadelphia chromosome? I just explained it. What happens is 9 and 22. So some portion of 9 gets detached and gets attached to the 22. You see over here, and some portion of 9 is broken and the 22 is broken, and it formed a new chromosome. I'm sorry, I think I explained it a bit wrongly. So what happened over here is a portion of chromosome 9 gets broken and a portion of chromosome 22 gets broken, and they both attach themselves like this. You can see, so they formed abnormal, and that is called as Philadelphia chromosome. And what happens in Philadelphia chromosome is, why is it common and to know about it is because chronic myelocytic leukemia, it is present in almost most of the cases of chronic myelocytic leukemia. So from that they came to know that if you're going to suspect CML in a person, you get it tested for this translocation. If the translocation is present, that means the person is going to develop CML. Okay, that is how its importance has increased. But you can see over here how the portions have got exchanged. Okay, now formal genetic disorders. These are some other kinds of genetic disorders which are present in the human being. One of the example is sickle hemoglobin. So what happens is we have hemoglobin of different types in our body. Okay, hemoglobin, when we are in the fetal stage we have fetal hemoglobin, and we become adult, we have adult hemoglobin. The difference is in the chains in the hemoglobin. So adult hemoglobin has alpha and beta chains, and the fetal has alpha and delta chains. Now what happens in sometimes, if the genetic disorder, some mutation has happened into the person, then instead of adult hemoglobin, some persons develop sickle hemoglobin. So HbS will develop. What happens in those individuals is they have sickle hemoglobin in their RBCs. The problem with the sickle hemoglobin is it can take on oxygen, but when it goes to release the oxygen into the alveoli, release the oxygen and take carbon dioxide, that's what hemoglobin does, right? It helps in the respiration, it releases the oxygen and takes in carbon dioxide. But in sickle hemoglobin it can take on the oxygen, but when it goes to releases itself in the in the alveoli, in the lungs, it will, when as soon as the oxygen molecule goes away, it gets changed, its shape changes from the biconcave shape of an RPC, it changes to a sickle shape. That's because it's an unstable hemoglobin. So it becomes a sickle shape. Now as soon as it becomes a sickle shape, it cannot take the carbon dioxide back in, and so then the vicious cycle starts. So oxygen is coming in, but the carbon dioxide is not going out, and because all the RBCs are not being able to conduct it, because as soon as they release oxygen, they get into the shape of sickle. So that is sickle hemoglobin, and those sickle RBCs are not good. They can form an emboli also, they can form anything, they can go in any part of the body and cause a problem. Okay, so that is a sickle cell hemoglobin. Most of the times people are just carriers. If they manifest disease, it's very difficult for them to live, because now again it depends how much gene is defective, how much gene is normal. Okay, how much of a proportion of sickle hemoglobin is present in individual, and how much of an adult hemoglobin is present. If you have only 2 or 3% of sickle hemoglobin and rest of all is adult, then you can live proper, I mean normally, right? But if you have like more than 30-40% of sickle hemoglobin and only 40% of adult hemoglobin, then you are going to have problems, right? So that is sickle hemoglobin. Second example is of phenylketonuria, where an enzyme goes abnormal, and so it will, so the phenylalanine, that is an amino acid of the adult, which is helps in the digestion of the metabolic process of the body, it does not go on. So the phenylalanine starts getting accumulated anywhere in the body, and the problem is that it gets accumulated in our brain also, and it can give serious problems or complaints like seizures, epilepsy and stuff like that. So the person will with phenylketonuria can have bad complications. Dominant allele and a recessive allele. Now what I'm trying to say over here is all the genes are shown by alleles. When they are dominant, they are shown by capital letters, and when they are recessive, they are shown by small letters. Okay, that is how they are symbolized. Now this is just to remind you about the pedigree chart. What is the pedigree chart? A circle shows a normal female, a square shows a normal male, a single bar indicates that they are husband wife mating, whatever. This shows a family, and this will show that it, a double bar shows that it was a consanguineous marriage. What do you mean by consanguineous marriage? That the marriage was between two relatives. It is more common than you believe. Many of the communities believe in consanguineous marriage, so cousins wed among themselves. Okay, so that's consanguineous marriage. This is like a fraternal twins. This is for the symbol for identical. If the individual is affected, you show it by coloring it. If the person is dead, you show it by slashing it. And if you're trying to show a carrier, you can show it by this, a circle and a dot. That is a carrier, and they are not going to manifest the disease, they only carry the defective gene, right? Now coming to the other types of genetic disorders are autosomal dominant, autosomal recessive. So what happens in autosomal dominant? If you have the dominant, you are going to show the disease. Okay, when does that happen? When there is germline mosaicism. What happens in this is there is a mutation in the germline of the parent cell. So parents will not exhibit the symptoms because they are already developed, right? But their genes have the problem, so the child is definitely going to develop the disease. Okay, that's one reason how the germline mosaicism or autosomal dominant can develop into a child. What are the examples of autosomal diseases? Are retinoblastoma gene, which is also a tumor suppressor gene, which we know it is an RBG which keeps a check on the cell cycle. Another example is in Huntington disease. Huntington disease is generally a kind of familiar disease, but what happens is it manifests very later in the stage. So late, so till that time the person has already had kids, right? So it has already had kids, so they know that okay, the kid will be like, okay, my mother has developed problem, I am definitely going to develop the problem, right? So that's Huntington disease. That's an autosomal dominant. And you look for the gene. What was the gene that was abnormal in the mother or the father because of which they develop Huntington? So if that you also carry the same thing, then you are also going to develop the Huntington disease. That's how Huntington autosomal dominant disorder happens. Okay. Then another example is type 1 neurofibromatosis. Again, it is a now all these autosomal dominant. What is another feature of these is it depends how much of a abnormal gene you are carrying. Okay, not everybody will be like, okay, you have 10% of defective genes, so you're going to have this much symptoms. It's not like that. You can or cannot also have the disease. Okay, that is a variation which goes on in the autosomal dominant disease. You have 10% of defective gene, so you might develop one or two complaints of the disease, and or you have only 10% of defective gene, you might not develop any complaints of the disease. Okay, that is the feature of autosomal dominant. Now what is autosomal recessive? Autosomal recessive disease start manifested because of the consanguineous marriage, because recessives do not manifest anything. They only manifest if both the parents are carrying the recessive genes, right? And the recurrence risk is like 25%. And the example of it is cystic fibrosis. So what will happen is mother will also be carrying a cystic fibrosis gene and the father will also be carrying a cystic fibrosis gene, and when both of them had a baby, so they had a baby with a both the defective genes which manifested the disease of cystic fibrosis in the body. Cystic fibrosis person will have a lot of problems. Okay, so their immunity is down, and because they don't have like a cilia or anything in it, so all the places where the movement should happen does not happen. So they will have, like even our respiratory mucosa is okay because of all the cilia, but the cilia is not working, so imagine so much of congestion. Then even in reproduction you have cilia which is trying to pass everything, eggs and sperms and everything. If the cilia is not there, you're not going to have all those kind of, you're not going to have it successful, right? So cystic fibrosis has very complicated problems. Cilia is also present in our respiratory tract. Basically cystic fibrosis patients have a lot of respiratory problems. From that we are jumping to some of the terminologies. So it is kind of like what is incidence rate. So it is the number of new cases a disease is reported during a specified period divided by the number of individuals in the population. So for example, we have 100 people in a population and 10 of them developed new disease, for say for example Huntington. So you will say the incidence rate of Huntington disease in that population is 10 by 100, 1 by 10, right? That's a very high ratio, don't think about it, but anyways, prevalence rate is the proportion of the population affected by a disease at a specific point in time. Okay, so I can say like at a specific time, so I can say during the year 2019, 25% of the people of the population of Toronto developed diabetes. So the prevalence rate of diabetes is 25%, right? Like that. uh, I got the percent by dividing the number of new cases with the population of Toronto. Okay, now relative risk is increased rate of the disease among individuals exposed to a risk factor divided by the incidence rate of the disease among the individuals who are not exposed to a risk factor. So relative, you are trying to find a relativity. What relativity? Like for example, you are trying to find out, you are studying lung cancer, you are trying to find out, okay, how many of the people who developed lung cancer were smokers and how many of them developed lung cancer were not smokers. So you found out the relative risk, how much is the risk of person who is smoking, how much is the risk that they are going to develop lung cancer, right? And this was very commonly done in India because there is pollution also which is contributing to lung cancer, so smoking and pollution and everything. So they tried to find out the relative risk of all these factors and how the people can develop lung cancer. Multifactorial inheritance. Now what does this term and why are we studying it is because some diseases are not because of only defective genes. They have many other factors which contribute, like there are environmental factors also. They can also cause a variation due to which the person can develop disease. That is called a multifactorial inheritance. It's not just one gene defect that is causing the disease. Okay, so what are those diseases which are multifactorial? So coronary heart disease is not just the one of the gene of the coronary artery is going problematic. The person also eating very much fatty food, the person is not doing exercises, there is so many other factors, the person is taking a lot of stress. All these factors contribute to the development of the coronary heart disease. The other examples are hypertension, cancer. When you're developing even breast cancer, it's not just BRCA1, BRCA1 or BRCA2 genes that are defective. There are other factors also, that the female is taking pills, OC pills, or female is having menstrual problems, a lot of stress, and all the other environmental factors. They can also cause breast cancer. Okay, colorectal cancer, diabetes mellitus, all these have multifactorial, not only just a gene going problematic, but also the environment around them which is causing problem in the individual. So diabetes, whenever we say, it is not just because the gene is abnormal in the individual is also eating a lot of sweet food, definitely. What are the other disorders which are multifactorial? Obesity, the gene and leptin gene is abnormal in them. There are dietary problems, there are exercise problems with them. Alzheimer disease, there are genes called presenilin 1, presenilin 2 which are going abnormal. Alcoholism, there is a gene which encodes for the ALDH2 which is defective, and because of that, this example I included is because of these individuals. Okay, why alcoholism I included is there is a gene ALDH2, we saw it in ethanol metabolism. Okay, some people do not have that gene, so these people, they do not metabolize alcohol. Because they do not metabolize alcohol, they do not get the effects, they do not get the symptoms of alcoholism. So they can drink as much as they want, they are not going to develop any complications, and that can lead to them becoming alcoholics. That's not good. But they will not develop all those complications. Okay, so those were all the multifactorial factors which were contributing to diseases. Now I have taught you before, I'm going to teach you again what are some of the epigenetic mechanisms which lead to those problems, those genetic defects, and also these epigenetic mechanisms contribute to development of cancer. So the examples are DNA methylation. What happens in simple terms is, DNA does not like anything foreign to it, but if a methyl group comes and attaches itself to the DNA, it is going to go crazy. So that's DNA methylation. Heterochromatin means a structure of DNA is always very tightly wound. It is always tightly wound around in the histones, and it is in a condensed state, not an actively transcribing stage. Okay, so always DNA material in our body is in a heterochromatin state. But if it becomes active, then it is, if it becomes abnormal, it becomes active, then it is called a hyperchromatic stage. Okay, that is a hyperchromatic stage in which the DNA can become active, and it becomes hyperchromatic because of any other problems like getting methylated or something like that. Okay, what is a histone? Histone is the positively charged protein around which the negatively charged DNA molecules are wound, which facilitates in DNA becoming very compact or tightly compact around each other in the nuclei. Set of histones and the segment of DNA wound around them are together known as individual nucleosomes. So this is just a terminology that you should know. What is a nucleosome? The histone and the DNA all together is known as a nucleosome. The mechanism is, we talked about it, heterochromatic. Euchromatic is when the DNA loosely bound to transcription factors are able to access it. So when the cell is ready to replicate, ready to multiply, it will become euchromatic. So the segments of DNA start getting loosened up. So when they start getting loosened up, it start gets separating, and because of the separation, the new strand can start getting formed. What are the histone modifications which can lead to the strand getting separated? Either the histone can also get methylated, a methyl group will get and attach itself to it, or it can get acetylated. Either way, even histone does not like a foreign thing on it. So if an acetyl group comes and attaches itself to the histone, the positive charge of the histone will decrease. As soon as the positive charge decreases, the two strands of DNA starts going away from each other, because histone was keeping it together by the positive charge, right? Now the positive charge has decreased, so the negative charges of both the strands starts becoming strong and they will repel each other. So that is histone acetylation. Histone methylation is when the methyl group comes and attaches itself, and it can work in either way. It can either keep the DNA very tightly bound, or it can make it loose.